Hello everyone, I'm Salma Sohrabi-Jahromi from Max Planck Institute for Biophysical Chemistry, and today I will talk about multivalent binding in RNA binding proteins. And before I start, I would like to thank the organizers for giving me a chance to present my work, as well as our amazing reviewers at the proceedings track for giving us very nice and constructive feedback that also leveled up this work. So in today's talk, I will introduce you to our tool called Bipartite Motif Finder, or BMF, that models bipartite binding of RNA binding proteins and searches for multivalent binding modes and results in motifs that look like this with the sequence preferences as well as the distancing between the two motif cores. I will show you how the tool works, and also I will summarize how it performs in comparison to other tools and also how well it can predict new binding sites in RNA. So the reason we were interested in multivalent binding modes was the observation that more than half of the human RNA binding proteins do have more than one RNA binding domain, and that is often of different types of RNA binding domains. And this is also pointed out before by other groups that this results in the identification of multiple sequences by these RNA binding proteins. So each domain can recognize one sequence, and these sequences can be spaced out within the RNA lengths. So this motivated us to design a tool especially or specifically for that, which we called, as I said, a bipartite motif finder. And we considered a case where the RNA binding protein has two domains, domain A and domain B, and with each of its domains can bind a sequence of length k, which we model through the binding energy of each domain to each of the possible k-mers. We also take into account the geometry of binding, meaning that when domain A has bound to the target RNA sequence, domain B is at the close proximity to the RNA strand and therefore has a much higher concentration. We can thermodynamically model this. And we also take into account that the concentration of domain B depends on how further away domain A has bound to the sequence, and we include in our model this distance-dependent concentration. So as a result, our model has 4 to the power of k energies for each of the domains, as well as the concentration parameters, which we model through a negative binomial distribution. And if we plot these parameters, we get something like this. So these are the energies which are translated into probabilities for each of the domain cores, as well as how well they can be spaced from each other. And I want to point out that while theoretically we have 4 to the power of k parameters for each of these models, in practice, the distribution is very biased towards less than a handful of k-mers, meaning that the effective number of parameters for the model is below 10. And this is also true if we increase the k from 3 to 4 or 5. So how do we learn this model? We learn it by looking at bound and unbound sequences derived from experimental data sets such as clip-based techniques or in vitro data such as high-throughput selects. And we optimize the model parameters in such a way that we have high binding probabilities for bound sequences and low binding probabilities for unbound sequences. So how does this work? To find the best BMF model, we have to calculate binding probabilities for each sequence in the dataset. And this is not so simple because this requires us to iterate all different combinations of one or more proteins binding in the sequence with different spacing between the domains. Luckily, we can solve it efficiently using dynamic programming, and we use it by breaking down this big combinatorial problem. So in our case, it means that if we know the probability of binding to the subsequence up to I-1, here shown in gray, we can easily extend it to calculate the binding probability of a slightly bigger sequence up to position I, which is just one nucleotide longer. And I'm saying it's easy because we know at position I what different configurations can happen, which is either nothing is binding, or domain A binds, or domain B binds as a complete new binding event, or a domain B binds whose domain A has been bound to the sequence upstream. However, there's a challenge, and that is the fact that for calculating the probability of the last configuration, we need to know the distance between the two domains, because based on the distance, the probability differs. So we have to somehow keep track of domain A throughout the dynamic programming. And we do this by splitting the configurations in two and iteratively calculate their individual probabilities. The first type of configurations, called A configurations in our model, are those that domain A binds last in the sequence. The rest of them we call B configurations, which are either those that nothing is binding or domain B is binding last in the sequence. And this is how we calculate the probabilities for each of these configurations. For the A class, calculating the probability up to position I means that we sum up all configurations up to position I minus k, their probabilities, and then multiply that with the probability of the current domain A binding, which is, according to Boltzmann's law, its concentration to the exponential of its binding energy. It's similar for B configurations, with the exception that we can have a couple of different things happening. Either nothing new is binding, so we fall back to the probabilities up to I minus 1, or we have the domain B binding, which completes the binding of domain A. So we have here the concentration of domain B, which depends on the distance. Or we have a domain B binding as a new binding event, which is similar to what we had before. The probabilities up to I minus k times the Boltzmann's law, which shows the probability of domain B binding. What I should also say is that these are not really probabilities, these are statistical weights, but they can be normalized to probabilities because they're proportional to them. Okay, so in this whole picture, the model parameters, BMF parameters, are the E's, so the energies of binding for each of these domains, and the CB, which defines the distance-dependent concentration. So we derive how the distance distribution is best based on the CB parameter. Okay, so how can we actually learn these parameters? How do we do it? We do it by stochastic gradient descent. To be precise, we use the Adam algorithm. And to show you roughly how it works, when I simulate data which is enriched in this type of motif, so we have AAA or AAU for the first k-mer, CCC or CCG for the second one, and such a distance distribution between them, and random sequences as a background, I see that the log likelihood increases as we expect throughout the optimization in each iteration, and then plateaus when the model has been learned. And we know that because we can actually track the energies of each of these k-mers for the first and second core. And we do see that the 4 k-mers which we expect to be enriched do constantly go down in their energies, and then they plateau, in contrast to the rest which are not bound, which increase in their energies. And the optimal distance distribution between them, which is around 5 nucleotides, is also learned throughout the iterations. So if we extend this approach to a couple more of implanted motifs, we do see that BMF consistently retrieves the same motif back. So it can learn both the distance preferences as well as the sequence preferences implanted in the dataset. And an important feature of BMF is that it uses a negative binomial distance distribution model, meaning that it can learn both higher distance preferences, as we see on the top, as well as just longer sequence preferences. So those which are not bipartite, but are just made up of a long stretch of nucleotides. And this is because it can put all of the probability mass at distance 0. So after proving the proof of principle that BMF can work, at least in simulated datasets, I moved on to try BMF on real sequences. So to do that, I looked at the high throughput select data for 86 RNA binding proteins, and I'm showing you the BMF results for 4 different RNA binding proteins in this data set. We do see that for some of these proteins, we have bipartite binding, showed by the higher distance preference between the two cores. And for some others, we just see the binding to a longer stretch of RNA. To quantify how often we see each case, I looked at the distance distribution, the proportion of it that falls beyond distance of 0, and I quantify how often we see that. And we do see kind of two extremes in most cases, being that either the whole distance mass is at distance 0 for some of these proteins, and for some other ones, the whole distance distribution falls for distances that are beyond 0. And these are roughly half-half. So half of the proteins in the data set do show bipartite binding. And another interesting thing which we can also see here just by taking a look at these logos is that when we have bipartite binding, often the sequence preference is similar between the two cores. And to quantify that, I looked at the correlation of the probabilities between the two domains for each of these cases. And indeed, it does seem to be that for bipartite motifs, the Pearson correlation between these probabilities is quite high compared to adjacent cores, which do show a very low Pearson correlation value. To evaluate the quality of these BMF models, I built a cross-platform benchmark that looks like this. So I trained BMF models on high-throughput select data, and then use these trained BMF models to predict new binding sites in ParClip and eClip datasets. I use this approach not only for BMF, but a couple of other tools that are out and often used in the field. So quantifying the performance with the area under the ROC curve results in a picture like this, where BMF models with a length of 3, 4, and 5 are shown in blue, and in red we have the GraphProt tool based on support vector machines and two deep learning tools, DeepClip and iClip. And we do see that BMF works a little bit better at predicting new binding sites, and this is also shown by looking at the logos of BMF when trained on high throughput select data and compared side to side with the logos we get when we train on clip datasets, as sometimes we not only see the sequence preferences being replicable, but also the distance preferences between them. So one last thing I want to show you is that we also made BMF available as a web server at bmf.zodinglab.org. So if we go to BMF's webpage, we see that we can submit a job by uploading enriched and background sequences in the format of FASTA, FASTQ, or plain sequence files. And if we just see an example results page, we do see that we provide an estimation whether or not the motif is bipartite, as well as the motif logo corresponding to the best BMF model found by our tool, and some stats on how the run was progressing, and also the final ROC curve on the training data. We also make BMF available as a command line tool, and the tool is open source and available on our GitHub page with instructions on how to install BMF, as well as information on how to run the main BMF commands, namely finding or training BMF models, plotting them, and using them to predict binding in new sequences. So to summarize my talk, I introduced you to BMF, or bipartite motif finder, as a tool for learning bivalent binding preferences for RNA binding proteins. I showed you how we estimate that half of at least the more studied RNA binding proteins do have multivalent binding. and showed you that BMF has a competitive cross-platform performance in predicting new binding sites. And I also introduced you to BMF web server that hopefully simplifies its application for new users. So this brings me to the end of the talk. First of all, I would like to thank Johannes for giving me the chance to work on this amazing project and for all the great scientific discussions we had. I'm also very thankful to Christian for his scientific and technical advice, as well as for introducing me to web server development, and to Milot for the technical support and feedback. And of course, to all the wonderful members of the Zoding Lab for the great working atmosphere and the insightful scientific and philosophical exchanges that we had. And I'm also thankful to DFG for financing my PhD, and to all of you for listening to my talk, and I'd be very happy to take questions now.